Welcome, everyone. Today, we're jumping into some really cool neuroscience. So let's put on our detective hats because we're about to crack a biological mystery I'm calling the case of the silent neuron.
All right, let's start with something I bet you've experienced. You ever leave a loud concert and everything just sounds muffled? Kind of like the world's volume knob got turned down? Well, what if I told you that feeling isn't just your ears ringing? Nope, it's actually a really clever, intentional process happening deep inside your brain. So, what's really going on?
So, first up in our investigation, we have to look at the central clue. That muffled feeling you get? It all starts with a period of, well, mysterious silence in the brain. Okay, so deep inside your brainstem, you've got these incredible neurons that process sound at lightning speed. But when they get hit with a ton of noise, like at that concert, they fire off a crazy burst of signals and then they just go quiet. For a little while, they just stop responding. And scientists naturally were desperate to figure out why. What's causing this sudden shutdown?
This right here, this is basically the electrical crime scene. On the left, you see those sharp, rapid-fire spikes? That's the neuron going nuts during all the noise. But look what happens right after. See that dip? The electrical signal actually drops below its normal resting line. That dip is the period of silence. The neuron has basically been forced into a cooldown, making it way harder for it to fire again. This is the mystery we need to solve.
So, what is this cooldown really? Let's get into the nitty-gritty and break down the anatomy of this silent period.
The fancy scientific name for this is posttetanic hyperpolarization, or just PTH. Posttetanic is just a way of saying after a ton of firing, and hyperpolarization means the neuron gets even more negatively charged than normal. Think of it this way. To fire, a neuron has to get from its resting state to a positive number. But during PTH, it's been pushed into the negative, so it needs a much bigger push to get going again.
And here's the thing, this isn't some random glitch. The length of this silent cooldown actually helps our brain figure out how loud a sound was and for how long it lasted. This timing is absolutely essential for one of the coolest things we do, pinpointing where a sound is coming from. So this isn't a bug in the system, it's a critical feature.
All right, so what's causing it? Every good mystery needs a prime suspect. And our first one is a tiny, tireless little machine inside the neuron.
Let's meet the sodium-potassium pump, or NK-A for short. The easiest way to think about it is like the cell's bouncer. After a neuron fires, all these sodium ions rush in. The NK-A pump's job is to kick them out and pull potassium ions back in to get the neuron reset and ready to go again. And after a massive burst of activity, this little pump goes into complete overdrive.
And this slide, wow, this is the proof. On the left, you've got the normal neuron activity, and you can see that big dick, right? That's our cooldown period. But then on the right, scientists used a drug to block that little NK-A pump. And just look, poof, the cooldown is completely gone. I mean, that is the smoking gun right there. It tells us that this little pump, when it's working like crazy, is literally what shoves the neuron into that silent state.
Okay, but if the pump pushes the neuron down into silence, what brings it back? You've got to have a counterbalance, right? Well, let's meet our second key player.
This is something called the IH current. So while that NK-A pump is busy pushing the neuron's energy down, the IH current is doing the exact opposite. It creates a slow, steady leak of positive ions back into the cell. It's basically a gentle reset button, slowly fighting the pump's effect and pulling the neuron back up to its normal resting state so it can get ready to fire again.
And here it is, straight from the researchers. They call the IH current a counterbalance to the action of NK-A. So you've got this amazing tag team. The pump starts the cooldown, and the IH current brings it back from the brink. It's this beautiful, delicate dance between two opposing forces.
So case closed, right? Well, not so fast, because the investigation took a really interesting turn when the researchers changed one simple thing that's often overlooked in the lab.
You know, a lot of this kind of research is done on brain tissue at room temperature, around 22 degrees Celsius or 72 Fahrenheit. But your brain isn't at room temperature, right? It's buzzing along at a cozy 35 or 37 degrees Celsius. So the scientists decided to crank up the heat and see what happened under more lifelike conditions.
And the result? It was huge. Just by warming things up to a normal body temperature, the part of the neuron that sends the signal got an 8 millivolt boost. Now, I know 8 millivolts might not sound like a lot, but for a neuron, that's like giving it a massive running start. It makes it so much easier to get ready to fire again.
This chart really shows you the payoff.
At room temperature, you get a bunch of spike failures, times when the neuron tries to fire during that cooldown but just can't make it. But at body temperature, those failures were cut in half. The whole system just got way more reliable and robust.
So what this all means is that temperature isn't just some small detail. It completely changes the game. At the warm temperatures inside our bodies, these auditory neurons are tougher. They fire faster and more precisely. And that silent cooldown period isn't quite so intense. It just goes to show, biology works best in the environment it was designed for.
Okay, let's wrap this up and give our final verdict. It turns out this whole period of silence isn't a problem with the system at all. It's a feature.
So we've found our three main players. You've got the Na pump, which kicks off the cooldown. You have the IH current, which hits the reset button and brings things back to normal. And then you have body temperature, acting like the master conductor, fine-tuning the whole performance to make it work perfectly inside a living brain.
Which brings us all the way back to that muffled feeling after the concert. That temporary quiet isn't your hearing failing you. It's an incredibly sophisticated, protective feature. It gives your auditory system a chance to reset. It prevents it from getting overloaded, and it helps it encode really important information about the sounds you just heard. It's a key part of how you make sense of the world. And that leaves us with one final thought. How many other things in our own biology that look like weird quirks or mistakes are actually brilliant solutions to really complex problems? It really makes you wonder what other hidden features are just waiting for us to discover. Thanks for helping crack the case today.